Hi everyone, welcome back to our biology class. Today we are going to discuss meiosis cell division. It is not required even though you have to see the video just for understanding. So let us start the video. Meiosis first.
Meiosis 1. In this module you will learn about meiosis 1. Meiosis is a nuclear division that occurs in the germ cells of sexually reproducing organisms during the production of reproductive cells called gametes. Meiosis is a reductional division that reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by two cell divisions, meiosis 1 and meiosis 2. The entire process of meiosis results in the formation of four haploid daughter cells with half the chromosome number as that present in the diploid parent cell. However, after fertilization occurs, the chromosome number gets restored. Meiosis 1 is the reductional division in which the chromosome number gets halved and distributed between two daughter cells. Meiosis 1 includes four stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 is the first stage of meiotic division 1. It is a very long phase and can take up to 90% of the total time required for meiosis. Prophase 1 can be further divided into 5 stages. These are leptotene, zygotene, pachytene, diplotene, and diakinesis. Let's know about the homologs before studying the phases of prophase 1. Each diploid cell has two copies of chromosome, one inherited from the male parent and the other from the female parent. These two copies, coming from different parents, are similar to each other and are called homologs. Now let us look at the different stages of prophase 1 in detail. To begin with, let us learn about leptotene. During leptotene, the chromosomes condense and become visible. Leptotene is followed by zygotene when the duplicated chromosomes pair with homologs to form bivalents with 4 chromatids each. This triggers the formation of a structure that holds the chromosome pairs together and promotes recombination. This structure is called synaptonemal complex. This process of attachment of homologs to each other along their length is called synapsis. The zygotene phase is followed by pachytene. During pachytene, the chromosomal crossing over now takes place between the non-sister chromatids of the homologous chromosomes. This crossing over is what makes each individual unique and causes the recombination modules to be visible. As the crossing over completes, it marks the end of the pachytene phase. The pachytene phase is followed by diplotene. During this phase, synapsis begins to break down and the chromatids start to pull apart. Due to this, the connections between the non-sister chromatids where crossing over existed become visible. These connections are called chiasmata. The appearance of the chiasmata marks the end of the diplotene. Diplotene is followed by diakinesis, which is also the last stage of prophase 1. During diakinesis, the chromosomes are fully condensed, the nucleolus and the nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase 1 is followed by metaphase 1. During metaphase 1, the microtubules from the opposite poles attach to the chromosomes at the kinetochores, and the bivalents align along the equatorial plate with a random orientation. Either of the parental homolog takes up any site on the spindle. The metaphase 1 is followed by anaphase 1. During anaphase 1, the homologous chromosomes separate out and move to opposite poles. With shortening of the microtubules, the sister chromatids, on the contrary, remain attached at the centromere. The anaphase 1 is followed by telophase 1, which is the last phase of meiosis 1. During telophase 1, the chromosomes reach the poles and the nuclear envelopes are reformed around the two daughter nuclei, and the nuclei reappear. A furrow appears in the cytoplasm and the cell divides into two by cytokinesis. The two daughter cells then enter a short interphase called interkinesis, during which there is no DNA replication. Therefore, at the end of meiosis 1, there are two daughter cells, each having chromosomes with two sister chromatids. In this module, you have left. Meiosis is a reductional division which reduces the chromosome number of a cell by half. Meiosis involves a single round of DNA replication followed by two cell divisions, meiosis 1 and meiosis 2. Meiosis 1 includes 4 stages: prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 is further subdivided into 5 stages: leptotene, zygotene, pachytene, diploidine, and diakinesis. During leptotene, the chromosomes condense and become fissed. During zygotene, the duplicated chromosomes pair with homologs to form bivalents. During pachytene, the attachment of homologs to each other along their length, that is synapsis, is complete. In diploidine, synapsis brings to breakdown, the chromatids start to pull apart, and chiasmata are visible. By diakinesis, the chromosomes are fully condensed, the nucleolus and nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase 1 is followed by metaphase 1, when the microtubules from opposite poles attach to the chromosomes at the kinetocourse and the bivalents align along the equatorial plate. During anaphase 1, the homologous chromosomes separate out and move to opposite poles, with shortening of the microtubules, forming two haploid sets. During telophase 1, the chromosomes reach the poles, and nuclear envelopes are reformed around the two daughter nuclei, and the nuclei reappear. A furrow appears in the cytoplasm, and the cell divides into two by cytokinesis.
Meiosis 2. In this module, you will learn about meiosis 2. Have you ever wondered how the chromosome number in humans is conserved generation after generation? Well, this is achieved by a type of cell division called meiosis. Meiosis is a specialized nuclear division in which the chromosome complement of a cell is halved. Meiosis takes place inside the gonads of sexually reproducing organisms and results in 4 daughter cells, all of which are haploid. Meiosis involves 2 steps, meiosis 1 and meiosis 2. Meiosis 1 is the reductional division in which the chromosome number gets halved. Meiosis 1 is followed by meiosis 2 after a very short interphase called interkinesis, during which DNA replication does not occur. Unlike meiosis 1, meiosis 2 is an equational division. In this module, you will focus on meiosis 2. Each daughter cell formed after meiosis 1 inherits only one of the 2 homologous chromosomes, and each homolog has 2 sister chromatids. Meiosis 2 involves the separation of these sister chromatids to produce haploid cells. Therefore, Meiosis II is an equational cell division. As both the daughter cells produced from Meiosis I undergo Meiosis II, the final result of Meiosis II is 4 haploid daughter cells. Like Meiosis I, Meiosis II can also be divided into 4 stages. These are prophase II, metaphase II, anaphase II, and telophase II. Prophase II is very short. During this phase, the nucleoenvelope and the nucleoli disappear and the spindle is formed with the help of the microtubules. In addition, the chromosomes recondense during the prophase II and become compact. Prophase II is followed by metaphase II, during which the chromosomes align along the equatorial plate and the microtubules attach to the kinetochores of the chromatids. Metaphase II is followed by anaphase II. In anaphase II, the sister chromatids separate out at the centromere and move towards the opposite poles as the microtubules become shorter. Anaphase II is followed by telophase II, which also marks the end of Meiosis II. During telophase II, the nucleoli are reformed and the haploid nuclei get enveloped in membranes. In addition, the chromosomes decondense and the spindle disappears. Division of the cytoplasm, cytokinesis, results in 4 haploid daughter cells. Meiosis plays a significant role in the life of sexually reproducing organisms. It results in gamete formation and helps conserve the chromosome number in sexually reproducing organisms. All the 4 gametes produced are genetically unique as they are produced by the crossing over of homologous chromosomes. The genetic uniqueness of the gametes is further attained by the independent assortment that results in each daughter cell receiving either homologue from each chromosome pair. Let's summarize what you have learned. Meiosis is a specialized nuclear division in which the chromosome complement of a cell is halved. Meiosis involves 2 steps, Meiosis I and Meiosis II. Meiosis II has 4 stages, prophase II, metaphase II, anaphase II, and telophase II. Prophase II is very short. The nuclear envelope and the nucleoli disappear, and the spindle is formed. The chromosomes recondense and are compact. During metaphase II, the chromosomes align along the equatorial plate, and the microtubules attach to the kinetochores of the chromatids. In anaphase II, the sister chromatids separate out and move towards the opposite poles. During telophase II, the haploid nuclei gets enveloped in membranes. The chromosomes decondense and the spindle disappears. Cytokinesis results in 4 haploid daughter cells.
Dear students, I hope you got the video. In this meiotic cell division, that stages are not required, only the significance of meiotic cell division is required. Notice that 2 points and learn it. What are the 2 significance of meiotic cell division? First one is it reduces the number of chromosomes into half. At the same time, maternal and paternal characters will intermix together during this phase. So these are the 2 significance of meiotic cell division. Learn only these 2 points, and also that the process of crossing over is also important. You learn that portion also. Thank you. I will come with a new video in the next class. Till then, bye.